Welcome, everybody, to the HAI Weekly Research Seminar. My name is Patrick Hines. I'm Senior Manager of Research Communities here at HAI, and really delighted to have everyone back. This is the first seminar of the academic year, and we are delighted to have Dr. Brian HAI with us today. Dr. HAI is an Assistant Professor of Chemical Engineering. He is the Dieter Schwartz Foundation SDS Faculty Fellow at Stanford University and an Innovation Investigator at Arc Institute. He supervises the Laboratory of Evolutionary Design, where they conduct research at the intersection of biology and machine learning. Dr. HAI was previously a Stanford Science Fellow in the Stanford University School of Medicine and a visiting researcher at Meta AI. Today's seminar, which is titled Genome Modeling and Design Across All Domains of Life, will introduce EVO2, a biological foundation model trained on 9.3 trillion DNA base pairs from a highly curated genomic atlas spanning all domains of life. We're thrilled to welcome Dr. HAI for this seminar and looking forward to his talk momentarily.
Thank you, Patrick, for the kind introduction. I'm excited to share some of the work we've been doing on developing genomic foundation models and then using them to generate new biology. And I wanted to start with this quote. This is from Frances Arnold's Nobel lecture, actually, where she says, "Today we can, for all practical purposes, read, write, and edit any sequence of DNA, but we cannot compose it." And the idea is there's been tremendous advances in our ability to obtain the sequences of DNA, write those base pairs, and actually do genome editing. But it's still hard to actually compose completely new DNA sequences because, in part, biology is completely unintuitive to us as humans, and it's in this foreign language, right? So here's a cartoon of what biologists like to describe as the central dogma of molecular biology, where DNA encodes for RNA, which encodes for protein. And at each step in this code, it's completely unintuitive to me, and I look at protein and DNA sequences almost on a daily basis. So how do we actually teach models to extract higher level information from this unintuitive code? So over the past few years, there's been tremendous advances in machine learning models that can learn higher order rules and grammar and semantics and other things from raw sequence information. And fundamentally, these models are trained with very simple objectives, like predicting the next token in a sequence of tokens, where a token could be an English word, for example. So in this first sequence, to be or not to, probably everyone could complete the next word with be if you've read Shakespeare. But GTG, CAT, CT, I don't think anyone could guess the next base pair in this sequence. But there are actually rules that might allow a model to learn what the next base pair actually is. And the idea is that even though next word prediction in language seems like a very simple task, potentially this task actually encompasses many complex tasks within it. So a good next word predictor in language would have to have some understanding, or at least simulate some understanding of grammar. For example, in my free time, I like to code would be a valid completion, but in my free time, I like to banana would not be. But also a model would have to learn semantics. So for example, I went to the store to buy papaya, dragon fruit, and durian could be a valid completion of this sentence. But squirrel might not be, even though it's a noun. It's probably not something you could buy at a store, for example. But also the model would learn facts about the world, or how words are associated with human sentiment, or translation between languages, or spatial reasoning, or math. Eventually, all of these concepts are embedded in text from the internet, for example. And in biology, we typically like to think of these two kind of domains. There's genotype, which is the DNA sequence, and then there's phenotype, which is the actual biological traits that we observe in nature. And this genotype will encode for phenotype via some complex mechanism. And it's often these mechanisms and phenotypes that we want to understand in biology. But in biology, it's also very hard to collect a lot of data at the level of mechanism and phenotype. But it's actually also very easy in biology to collect data at the level of genotypes through advances in next generation sequencing. Actually, the amount of genomic data outpaces data generation from the internet. So there's a lot of DNA sequencing data. And potentially, we could leverage the fact that even though evolution acts on phenotypes, the results of evolutionary selection get written back into genotype or the sequence information. So in the same way that large language models don't have an explicit world model in them, enough about the world has left its imprint on text from the internet. And in the same way, in biology, the world has left its imprint on DNA sequences through natural selection. So a potential hypothesis could be that we can teach a DNA language model to predict the next base pair, and this could teach it things about which mutations are associated with disease or protein biochemistry or RNA structure or even higher level systems biology. A lot of people have been doing this, not for DNA sequence in biology, but for protein sequences. So for example, you can train so-called protein language models on sequences of amino acids. And I won't go into too much detail in protein language models and related models in this talk, but suffice to say that these models have really revolutionized protein biology. So for example, last year, the Nobel Prize in Chemistry was awarded to methods for protein structure prediction, like AlphaFold, if you've heard of that model, or methods for de novo protein design from the group of David Baker. And we are still very interested in protein design and generating new proteins, but we're also interested in how these proteins come together to make, say, a complete organism. But when you look at it, even a very simple single-celled organism is orders of magnitude more complicated than any individual molecule. And we want to have potentially machine learning models that can understand this complexity and even design new biological systems at this level of complexity. So how would we even do this? How could we, say, improve the level of our generative models to achieve whole organism design? And that's what I wanted to talk about today. I'll talk about various machine learning, frameworks or ways of training and using machine learning models to get us towards, say, whole genome design. And the first thing I wanted to talk about is something called unsupervised pre-training. And for those of you who are unfamiliar with this jargon, one way I like to think of this is, who would you rather train let's say you're a wet lab biologist and you need some help in the lab. Who would you rather train to help you pipette in the lab? Like, would you rather train a high school student or would you rather train a baby? And most of us would probably pick the high school student because even though both of these people don't have, they would not know how to pipette at the beginning, the high school student has still acquired, fine motor control or the ability to speak in language. And these are unrelated to pipetting, but can still transfer to the task of pipetting, whereas the baby would have to learn from scratch. So similarly, we would like to start with a model that has some information about very complex biological concepts. And for this, we can leverage the fact that for the past 30 decades now, people have been sequencing the complete genomes of organisms and just depositing these genome sequences in publicly available databases. And in theory, embedded in that DNA sequence, data is the information needed to create whole organisms. So we want to extract this information from this raw, unstructured data in these databases. So around early last year, we put out this model that we called EVO, or EVO1 for the sake of this talk. And it's a DNA language model. And it's actually doing a very simple task. All this model is doing is it takes in a DNA sequence and predicts the value of the next base pair in the sequence. This model is trained on prokaryotic genomes. So these are very simple organisms like bacteria and archaea. But the idea for this proof of principle model is that even though we're only training models on DNA sequence, because of the beauty of how biology encodes information, DNA sequence actually just directly maps to RNA sequence, and DNA sequence will also, through RNA, directly map to protein sequence as well. So one important idea is even though we're only training a DNA model, we potentially get RNA and protein modeling for free. Another important idea behind the original EVO model is that the model is trained on raw genome sequences and can process an input sequence much longer than any single gene. So EVO1 could process an input sequence of 131,000 base pairs. And at this scale, you not only get information about, say, a single protein or a single gene, but how that gene interacts with its genomic context at the scale of maybe multi-gene systems, and for some very simple organisms, the complete genome in the context as well. EVO1 was, though, only limited to prokaryotic genomes. So these are the genomes of bacteria and archaea, these two domains of life. There's a third domain of life, the eukaryotes, and this is an important domain of life. It includes plants and animals, and humans are eukaryotes too. So we wanted to expand this modeling paradigm beyond the prokaryotes to the eukaryotic domain as well. The challenge is that eukaryotic genomes are much more complex than prokaryotic genomes. They're billions of base pairs long instead of millions. There's very complex genetic architecture, like the genes are not contiguous on the sequence. They're often like, there's large gaps in between a given gene. And so this required a lot of work and data processing and scaling up the models to actually bring this modeling paradigm to this third domain of life, the eukaryotes. But we did this, and so this is a model that we put out earlier this year, which we're calling EVO2, and this is a genomic foundation model trained across all domains of life, including the eukaryotes. So each dot on this slide is a different organism's genome that we have in the training data. And so we train this model from bacteria all the way to humans, but we also include insects and plants and other things. This model also has, it can process an input sequence now of up to 1 million base pairs. We scaled this model to 40 billion parameters and trained it on 9 trillion tokens, where each token is a single DNA base pair. We did this with the help of NVIDIA, who graciously gave us 2,000 H100 GPUs to train this model. And we think that this is a very useful model for not only predicting biology, but for generating biology as well. And so the generative tasks are what I want to focus on today in this talk. So once you have a model like EVO2, how do you actually use it to generate sequences? And we basically use the fact that this model can predict the value of the next base pair in the sequence. So say you sample the first token, which is an A in this case. You can use the value of the first token to predict the value of the second token in the sequence, right? And you can use the first 2 tokens to predict the next token and kind of auto-regressively sample a new base pair each time. And what we did with the original EVO models is we first wanted to see what happens if we just did this, but like sampled 1 million tokens. And we thought that sequence would be completely garbage or would quickly degenerate into repeats or complete white noise. But actually, with EVO1, the generated sequences at 1 million base pairs actually looked somewhat reasonable when we analyzed them with traditional computational biology like heuristics. So there's predicted genes on these sequences. We actually find tRNAs that cover all 20 canonical amino acids. So in some way, these genome scale designs looked somewhat reasonable, but in other metrics, they're obviously not going to be real genomes. We liken this to the first versions of image generation models where they would produce say like a blurry image that looks somewhat reasonable, but obviously not like a photorealistic image. With additional pre-training, the model can generate less blurry genomes. So for example, with EVO2, when we generate things, prokaryotic genomes at the scale of hundreds of thousands of base pairs, the coding density looks a lot better. The proteins look a lot more natural, but we still think that these genomes would not function if we actually put them into nature. And there's still a lot of work to do on our kind of roadmap towards whole genome design. Okay, so what is the second strategy that we can use to improve the generative quality of our models? And this is something called supervised fine-tuning. Intuitively, if we take that high school versus baby analogy, this is like actually now teaching the model and specializing it on the task, putting in the 10,000 hours to learn the task. So for example, we can also do the same thing with these DNA language models. So we can take the EVO1 model in this case, And here we specialized it on around 70,000 sequences from a genetic system in nature called CRISPR-Cas. This is a system used for genome editing. It's actually a very complicated system. It has a very large protein that's 1,300 amino acids. And it also interacts with a non-coding RNA. So there's actually 3 different components, 2 different RNAs and a protein that all come together. And we train the model on a sequence with the protein and the 2 RNAs. So the model then can generate, when it sees actually a special token, the model knows to generate an entire what's called CRISPR-Cas operon. So all 3 of these components. We can actually see that we need a model to generate coherent sequence. We need a pre-trained model to generate coherent sequences. So if we just trained a model only on CRISPR-Cas sequences from scratch, the model still doesn't have enough information to generate complete Cas proteins, for example. But if we start with a model that already has some generalist understanding of biology and then specialize it to CRISPR-Cas, then these sequences start to look a lot better. One of the sequences generated by this fine-tuned version of EVO, this is a diagram of what the DNA sequence looks like. It contains a complete Cas9 protein, and we can detect the RNAs on the sequence. We put the sequence through AlphaFold, and the predicted structure resembles that of known CRISPR-Cas systems. We can then actually chemically synthesize the DNA and test it in the laboratory, and we compare against a state-of-the-art system for genome editing, SP-Cas9, which is from nature, and we show that our AI-generated Cas9s can cleave double-stranded DNA as well in vitro as SP-Cas9. But also this generated molecule has less than 80% sequence identity to anything in nature. For context, at this level of sequence novelty, we actually break any patents around CRISPR-Cas, and so these molecules are novel, at least in the eyes of the US Patent Office, for example. We can also use this approach, the supervised fine-tuning approach, to design these systems. I won't go into too much detail, but they're proteins that bind to DNA, and these proteins can also hop around in different from one genomic position to another. So we are actually able, with supervised fine-tuning, to generate coherent biological systems that work when we test them in the lab. But there are some limitations to this, like it requires a lot of data and some compute to train the model. With naive applications of supervised fine-tuning, you might also elicit some memorization or recall from the distribution that you use to actually fine-tune the model. So we wanted to see to what extent we can then prompt the model or otherwise elicit not trivial memorization, but actually elicit some amount of creativity and novelty. And so for this, we use a technique sometimes referred to as prompt engineering or sequence conditioning or context engineering. And to give some intuition for how this works, here are 2 different prompts that you might see on, say you're sitting in science class, for example, and you see 2 different prompts. One prompt is provide the balanced chemical equation for oxygenic photosynthesis, then name the 2 major stages of this process, specifying the primary inputs and energetic outputs for each stage and their precise locations within the chloroplast. I don't know if anyone here could actually answer this. A second prompt could be, what if humans could photosynthesize? And in one prompt, you might actually elicit a lot of memorization and recall from the person that you ask. The other prompt might elicit more creativity and have a bigger diversity of potential responses. Okay, so with this idea, we actually then wanted to see if we could prompt the model with DNA and actually elicit some creativity from the model. And for this, we actually leveraged the fact that the EVO model, when it's trained on prokaryotic genomes, it's trained on multiple genomes in the same context. And actually, a nice thing about prokaryotic genomes is that genes with similar functions are also next to each other on the actual sequence of the genome. Or genes or genes with related functions or genes that inhibit other genes, they're often next to each other on the genome sequence. So for example, we can prompt the model with a gene where we know its function, and then have the model generate a completely new DNA sequence, and then see if there's a gene in that generated sequence that has a similar function or a related function to the gene in the prompt. So when we do this with these highly conserved genes, so these are gene clusters that don't change very much across evolution, we get memorization from the model. And so, for example, we can prompt with mod A, which is a very conserved gene, and get mod B in the response as expected. We can prompt with mod B, get mod C in the response. This is not that interesting, but it just shows that the model can respond to DNA prompts in its input. So now we actually want to prompt the model to generate things that are very evolutionarily novel. And for this, we focused on a system called anti-CRISPRs. So these are proteins or genes that inhibit CRISPR. So for example, they bind to Cas9 and inactivate it. And actually in nature, these anti-CRISPR genes are very novel. And this is related to their biological function. They're part of the arms race between bacteriophage and bacteria. If you have something highly conserved in this arms race, you're not going to survive. You need a lot of novelty, evolutionary novelty, to just keep up. And so actually, these anti-CRISPR proteins are very novel. A lot of them are even hypothesized to emerge from de novo gene birth, from nothing to a gene. And these anti-CRISPRs are also often associated or next to a gene like a gene called an anti-CRISPR associated gene, or an ACA gene. So we can actually prompt the model with an anti-CRISPR associated gene and see if in the response we get anti-CRISPR proteins that are novel. So we did this with the model, and we can actually detect genes in the response that when we test them in the lab, they do have anti-CRISPR activity. So in this case, they can inhibit Cas9 and inhibit CRISPR. So they are experimentally confirmed to be anti-CRISPRs. But then we did an interesting experiment where we then looked at the sequence of this gene and compared it to all of the gene sequences that we've observed in nature. And actually, for a couple of these genes, we can't detect any significant sequence identity above, say, a random null to any gene that's ever been discovered from nature. And so we think that this is strong evidence that the model is capable of actually generating things that are quite novel and being creative in some extent, and is not, it definitely can memorize and recall its training data, but if prompted in the right way, the model can also generalize and produce sequences that are still functional, but in terms of the actual sequence composition are unlike anything that we've seen in nature. We then want to do this prompting strategy beyond just generating anti-CRISPRs. So we actually generated this database we called Syngenome that has 100 billion base pairs of AI-generated genomic sequence spanning thousands of putative functions. Okay, so this brings us to the fourth technique in our arsenal for improving the quality of generated sequences. And this is something called, or I'll refer to in this talk as like inference-time guidance. And the idea is that in some cases, the model, if you want to push the model into a regime that is totally unnatural, that's not like anything in its training data, you might be able to still leverage a lot of the prior information learned by the model, but then couple it with other models that can push it into areas that are kind of outside of its training distribution. Okay, so here is one example of how we can steer the model's outputs with a separate scoring function. So here we have, it'll take a long time to explain this task, so bear with me. But in the genomes of mammals, humans definitely, and mice, for example, certain parts of the genome sequence are more or less accessible or open to proteins to bind. So some parts that are very inaccessible, proteins don't bind, not a lot happens in those sequences of DNA. In other sequences of DNA, they're very open, lots of proteins can bind and maybe turn on different genes. So we wanted to see if we could control the openness or the closedness of DNA. This is part of epigenomics, actually, in human genomes. We wanted, though, to design sequences that were very unnatural, just to show that we can control which regions of the DNA were open and closed completely synthetically. So our design task here was to design or to write Morse code into chromatin or into accessible DNA. So a Morse code dot would be a narrow region of accessible chromatin. A dash would be a wide region of accessible chromatin, and the spaces are just the closed chromatin. So EVO2 does not have any native understanding of which regions of a genome are more or less chromatin accessible. So we use these models called Informer and Borzoi in this case, and these are models that actually take in a DNA sequence and then predict how accessible that sequence is. So, and with these models, we can actually take EVO2 because it's a generative model. Informer and Borzoi are not generative models. They just predict chromatin accessibility from sequence. So we can, for example, sample a couple hundred base pairs from EVO2 and sample multiple of these sequences. We can then score them with Informer and Borzoi, and we can accept the sequences that have desired chromatin architecture. We reject ones that don't, and then append the ones that we accept to the prompt and continue sampling off of those kind of trajectories. And in this way, it's actually just a very simple beam search algorithm. But we can use this very simple algorithm to steer the model's generations to have the desired chromatin architecture that we want. Okay, that's very technical, but you can maybe have an intuition of how this is working in this slide. So on the x-axis is the position into the genome sequence that we're designing. On the y-axis is the predicted chromatin accessibility, and the bold yellow line is the Morse code pattern that we want to design. I'll leave it as an exercise to the reader what message we're actually encoding in Morse code, but a narrow peak is a dot, a wide peak is a dash. The white lines are the model's predicted design chromatin, and each frame in the GIF is a new iteration of beam search. So and it's actually the accepted sequence. So in this case, you can actually kind of see the model writing the chromatin architecture from left to right as this very simple beam search algorithm progresses across the sequence. So we have these designs that have the desired predicted chromatin architecture. We can then, again, experimentally test these designs and see if they have the chromatin architecture in the lab. For this, we partnered with a biotech company called Anza Biotech. They specialize in long DNA sequence assembly. So they helped us synthesize and assemble 20,000 base pair sequence chunks that we then delivered into a very specific location in the mouse genome. So these are mouse embryonic stem cells. We then let the cells rest for a little bit and then actually do this assay called ATAC-seq, which measures how open or closed the chromatin is. So this is what we do to validate our results. And we can see we actually get very good agreement between our computationally predicted designs and the experimentally tested chromatin accessibility that we see in these mouse cells. So we can actually, with pretty good control, use EVO2 combined with other algorithms at Inference-time to generate new regions of a mouse genome with the desired epigenomic patterns. More broadly, we think that this general paradigm of coupling a very capable generative model with an application-specific scoring function or even a reward function is a powerful paradigm for designing biological systems with desired properties. Okay, so I've talked about all of these different strategies for improving the quality of biological generations. Can we now put it together to enable whole genome design? And for this, I'll be talking about a recent work that we published, or we preprinted actually a couple weeks ago, led by Samuel King, who I think I saw in the audience, so you can find him after if you're interested. And this is work using EVO to generate complete genomes of these biological systems called bacteriophages. So they're viruses that infect bacteria. Phages and genome design in general is a very complicated design task for biologists for a number of reasons. You know, in a genome, you have multiple genes. they all interact with each other. They interact with genes that are not in the genome. There's also regulatory elements that are not genes, but they control gene expression and things like that. And so we wanted to see if these models can now go from designing one gene or two genes binding to each other to now designing things at the scale of an entire genome. Another reason why we picked bacteriophages is that they're an emerging therapeutic modality. And so, for example, this is a famous case from a couple years ago. This man, Tom Patterson, almost died from a multi-drug resistant bacterial infection, but actually his life was saved by phage therapy. And so he actually made a full recovery, and this is his wife is actually holding an image of the phage that saved his life. And so our design task in this case was to design phage that used as a design template this phage called PHI-X174. It's a very well-studied phage in molecular biology. It actually has 11 genes. Some of these genes actually overlap on the DNA sequence. So you have the same sequence, but in a different reading frame. The same sequence encodes for two different proteins in some cases. Okay, so how do we actually then design variants of PHI-X174 at the genome scale? It's very complicated. There's 11 genes, multiple regulatory elements, interactions with the host. Well, we can apply all of the generative techniques that I've been talking about throughout this talk. So, for example, we start with the pre-trained models. So we start with the models EVO1 and EVO2 that we developed, that we pre-trained in the case of EVO1 on 300 billion prokaryotic genomes, in EVO2 on 9 trillion DNA base pairs. And so these models already have some baseline understanding of biological sequences. What is the start sequence of a gene? What's the end sequence of a gene? What do regulatory elements kind of look like? And then we fine-tune these models on around 10,000, I think 15,000 examples of phages from the same family as PHI-X174. It's a phage family called Microviridae. We, in our fine-tuning strategy, we actually are able to, number one, we make sure that when we fine-tune the model, the sequence of the phage always starts around the first token in the sequence. We also prepend these special tokens to the model so that when it sees the special token at inference time, it can then know to generate the sequence of a phage genome. So we fine-tune both EVO1 and EVO2 on thousands of examples of Microviridae phage genomes. We then do a little bit of prompt engineering with these DNA language models. So what did we have to do in terms of prompt engineering? Well, we actually observed that if the prompt was too short, then the model would not have sufficient conditioning to generate a phage genome that resembled PHI-X174. It would say generate some random phage genome or just generate some random DNA sequence if the prompt was too short. But if the prompt was too long, then the model would just recall the PHI-X174 wild type, the original genome, and not introduce any sequence changes. So actually, there's this kind of sweet spot where the prompt is long enough to tell the model to generate something that looks something like PHI-X174, but it's short enough to also not provide too much conditioning to the model and allow it to be a little bit creative and generate variations of this phage genome. We can also then apply inference-time guidance. So we actually combine the model to propose sequences with application-specific scoring functions. I won't go into too much detail because it's a lot of technical biological detail, but the two main constraints that we placed on the model's sampling protocol, one constraint is just that the genome had a reasonable genetic architecture, so we can detect genes that we would also detect in PHI-X174. This is actually a very difficult problem because the publicly available tools for identifying the starts and ends of genes would not work for PHI-X174 for a number of reasons, including the genes in PHI-X174 overlapping with each other. That was a big issue for developing these gene architecture scoring functions. The other is a constraint that constrained a protein on the sequence to be a protein that is associated with the host, the bacterial host that we wanted to target. So in this case, PHYX-174 is known to infect a bacterial strain called E. coli C. So we actually used sequences, the protein that determines the host range of PHYX-174. We constrained the model to generate proteins that also resembled that protein from PHYX. Okay, so once we have the model, we fine-tuned it, we can sample from it, we can do this guidance, in this case, fairly simple rejection sampling at inference time. Then we can actually, again, chemically synthesize the DNA. We can assemble the genome in a simple chemical reaction in the lab, and then introduce this DNA into E. coli C in this case. And actually, because in the natural life cycle of PHYX-174, there is a component of the life cycle that has like a double-stranded DNA circle involved. Now, if you actually put that, if you actually put the chemically synthesized DNA of PHYX-174 into an E. coli cell, that E. coli will start expressing the proteins in of PHYX-174, and you can reboot the phage from the DNA sequence. And we developed a fairly high throughput assay for testing the validity of these generated genomes, where if we saw that the bacteria were dying, then probably the phage was working. And we can see if the bacteria is dying based on how cloudy the actual liquid is. If there's a lot of bacteria, the liquid will be quite cloudy. But if the phage wasn't working, then if the phage wasn't working, the bacteria would be allowed to grow, the liquid would be cloudy. If the phage was working, the liquid would be clear. And then this helped us initially screen about 300 generated sequences of these complete PHYX-174-like genomes. So screening around 300 sequences, we actually identified 16 phages that we know are capable of being viable whole genomes. And they're actually quite evolutionarily diverse. Here is just an image showing where all the genes on these AI-generated genomes are located. Here's a phylogenetic tree placing the AI-generated phages alongside their natural counterparts. One phage genome even has less than 95% sequence identity to any phage in nature. And under some taxonomic thresholds, this does qualify as a new species of phage. So I don't know if there's different rules for AI-generated species now. We also determined the structure of one of these phages and actually saw interesting structural changes at the molecular level. So for example, this one phage that we're calling EVO536 has a much shorter protein, a much shorter DNA packaging protein in its capsid. Actually, another group had tried to swap out the PHYX-174 DNA packaging protein with this protein that, in this case, the AI model introduced into the capsid, and saw that swap would not lead to viable phage, so it wouldn't work. In this case, the model was able to successfully make the swap by also introducing compensatory mutations in the genetic background of the phage. So it was actually able to make this previously impossible swap occur by also changing the sequence context of this gene. Another interesting thing that we observed is that if we pooled these AI-generated phages together, then that mixture of diverse phages could rapidly overcome bacterial resistance. So these are strains of E. coli C that are resistant to the wild-type PHYX-174. And we see that if we introduce the cocktail of AI-generated phages, then within a few passages in the lab, these phages can recombine and evolve in a way to rapidly overcome resistance on the part of the host, which could actually be quite useful for phage therapy. For example, if you have just more diversity in your phage cocktail, natural or AI generated, then this could make this phage cocktail more resilient to evolution on the side of the pathogen. Cool. So in summary, these are, we used all of these techniques. So we used pre-trained models that were then fine-tuned and conditioned in the right way and sampled at inference time, guided by separate scoring functions to enable the first AI-generated genomes. And bringing this back to this initial quote by Francis Arnold, we see kind of this work as kind of completing the process. So we could read DNA, we could write DNA, we could edit DNA, and now with AI tools, we can also use, we can learn from evolution to also compose new DNA sequences. It's also a little bit fitting that PHAX-174 was the first DNA genome ever sequenced back in 1977. It was the first DNA genome ever chemically synthesized in the lab back in 2003, examples of read-write, and now we have DNA design or DNA compose. Cool. So in summary, hopefully I've convinced you that these models are able to generate interesting new biology in the same way that generative models have made tremendous strides in, say, image generation or code generation. They're also helping us design new biological systems too. So for example, we can design these de novo genes like these anti-CRISPR proteins. We can design these multi-gene systems like these CRISPR-Cas systems. We can design multi-kilobases of mammalian chromatin architecture with the desired chromatin accessibility patterns. And we can design sequences at the scale of complete bacteriophage genomes. All of this work we make publicly available. We document all of our experimental protocols, our computational protocols in publications. We make all the code available. Actually, for Evo2, we make that model fully open. So not only the model weights and the inference code for the model are available, we make the training data and the distributed training code publicly available and open source as well. So these are links to the models and references to the publications that I've been talking about and the preprints. Finally, this is work done by a very talented and large group spanning many different areas of expertise. We have people working on machine learning algorithms and novel model architectures. We have people working on machine learning engineering and scaling these models to, say, large distributed training configurations. We have computational biologists working on model evals and training data, people working on experimental biology and actually making these designs in the wet lab, which is non-trivial. This is a big collaborative effort across multiple institutions, including Stanford, but also Arc Institute and NVIDIA, and a number of other institutions as well. And finally, a lot of this work was also led and done by people in my lab with the support of HAI, which I'm very grateful for. And I think there's the Hoffman-Yee Symposium in a couple of weeks where I'll also be presenting some additional work from this. So if you wanted to, there's the talks don't overlap in content, and so or they do, but it's presented in a different way. So just wanting to bring that up. And finally, thank you all for your attention. I'm happy to use the remaining time for any questions. So.
I have a few related questions. One, could you talk practically about what it took to build the Open Genome data set, day to day? Two, how many orders of magnitude do you think are left until you hit kind of a data wall in terms of what's easily accessible there? And three, what you think the paradigm will be once you do hit that?
Yeah. So with the prokaryotic data, it's somewhat simpler. A lot of that data, we know it's biologically, it's functionally rich. We don't do that much other than we know that this is also, these are lessons from natural language. We know it's bad to have a lot of duplicated data in the training data. So we do a species level deduplication for the prokaryotic data just to make sure there's not a million different copies of the same E. coli genome that all differ by a single base pair or something. So that's what we do for the prokaryotic data. We do that for the eukaryotic data too, but we also filter out a lot of regions of the eukaryotic genomes that clearly do not encode for anything that's functional. They're more like structural regions of the genome, like in the middle of chromosomes or at the end of chromosomes. There's lots of just non-coding DNA that's not very information rich. We filter a lot of those areas out. But yeah, and then in terms of like how much data, we're actually not data limited at all in this area of biology. There's a lot of genomes that we were actively removing from the training data because like we couldn't even complete a single training epoch over all the data. And I think there's terabytes of genome data out there from prokaryotes, but also eukaryotes too that we just didn't train on. So there's no data limitation here. Yeah.
Hi, Brian. Great talk. It was a lot easier to understand than I thought it would be. Question. So you said that EVO2 has a context window of 2 million tokens, and you trained it on all kinds of eukaryotes, including humans. The human genome has 3 billion base pairs. So how did you get around that context window for longer genomes?
Yeah, so the model, we context extended up to 1 million base pairs. We definitely don't get the whole genome sequence in there. And so there's tasks, say, like if you wanted to understand how a gene on chromosome 1 interacts with a gene on chromosome X or something, probably these tasks are out of the scope of this model. This model is going to mostly give you information within a single gene in the human genome, like the gene and maybe some regulatory elements that are right next to it on the genome sequence. So in this sense, the model is still limited to 1 million base pairs. So we basically just don't even bother trying to fit all 3 billion base pairs into the model. We are interested in future versions or trying to consider what it would look like, or even if it would make sense to extend this, or how long we can extend this context and still learn valuable information, including potentially we would want future versions of genome models or other just completely different models to learn these long range interactions too.
Thanks for a great talk, Brian. So my question is, in language models, we've seen the next word objective contain a lot that's required in understanding language and reasoning, but it's not sufficient necessarily to understand the physical world, right? So people are now working on building world models. So I just wanted to ask in this context, like, what do you think next token prediction can achieve in terms of understanding of the genome? And what aspects do you think bringing in other modalities related to phenotype or environment will be required? And well, yeah, what directions do you see in kind of building multimodal foundation models with these kind of gene sequence models with these other modalities?
Yeah, it's a great question. I think like I also take a similar view, or I think these sequence models are a great prior. And it turns out in biology, a lot of biology is just getting onto the manifold, so to speak, or just like there's a lot of just DNA sequence that is not going to lead to anything viable. So the prior is very valuable in just getting you to a space within biology that's at least somewhat reasonable. But yeah, if you want like higher level control or a very specific function, you probably need to couple this very capable prior with a very like other specific predictive models, have it interact with the world and improve with experimental feedback. So yeah, I think it's actually fairly similar to other domains and in AI like robotics or these chatbots. Yeah.
Thank you. How much are you bottlenecked in the size of what you can reboot in the lab? Like generating a small phage, I know that was hard to even synthesize the DNA and get that to work, but how do you test the bigger predictions, like a bigger phage even, or a whole cell if we can't synthesize it, or can we?
Oh yeah. So I think, yeah, this is probably the biggest challenge. There's still a big computational challenge, but I think in terms of like how to get to AI generated life, for example, like I think the experimental bottlenecks are much more resource intensive than the computational bottlenecks. We can all like the model can generate 1 million base pairs in an evening, so it can generate the sequence. In terms of this, like Gibson or Golden Gate assembly of these phage genomes, this might get you to like a few tens of KB. for life, that's a few 100,000 or a few million KB. There you might need to like redesign things at the tens of KB level and do like a sequential replacement or something, or like confirm that a bunch of 50 KB sequences all work and then like combinatorially combine them together and then see if that thing lives. But yeah, I think that is an interesting question. There potentially be a lot of creative experimental ways to hopefully to be able to test the outputs of these models at the scale of like a living system. So that's actually the biggest bottleneck. Yeah. If you have any ideas on, I'd love to hear that. Yeah.
Oh, first of all, super exciting. It's almost godlike powers. You're able to now create life that's in a way that has never existed, because you mentioned some of these things have never come up through evolutionary biology. So I had a twofold question related to responsible AI. And so one is, are there some risks with some of the quote unquote life forms that you're creating that have never evolved over billions of years, that they could actually become very harmful in the environment? And is there anything that you're able to do for that? Maybe the current EVO2 model isn't able to produce that, but it's obviously the direction that you're going with whatever EVO3, 4, 5, whatever, it might be able to create whole cells. It might be able to create whole organisms that could be so novel that the rest of biology doesn't know how to react or how to deal with them. And then number two, it's great that you're open sourcing essentially everything, the weights, the data, and everything like that. Are there any risks that it could be used for harmful purposes as opposed to for good that you're also worried about?
Oh, yeah, this is a really great question, and we try to be very thoughtful about safety and containment of the systems that we generate. So I guess there's like maybe two tiers of responses, like what the models can immediately do and what we've done already. But maybe I can also try to speculate on the future of these technologies. In terms of like what the models can do, for example, for the phage work, we're very careful in terms of our experimental protocols. We're testing phages that we understand quite well. And they're being tested against a non-pathogenic strain of E. coli. And we're taking additional precautions in the lab so that like everything gets disposed of as biohazardous waste, for example. So we do take lots of caution around containment and safety, though a lot of those safeguards are built into the non-pathogenic nature of the system. We also did a lot of thought on when we were developing the original EVO2 model on everything that you mentioned. And for this reason, the EVO2 model is actually not pre-trained on any eukaryotic viruses. So these are viruses that infect everything from yeast to humans. And so and we can actually empirically test that the model can't generate, that the pre-trained model can't generate eukaryotic viruses or score any properties of them very well. We decided to open source the model because we think that AI is safest when it's out in the open. If say you wanted to fine tune these models on eukaryotic viruses, that would be a non-trivial effort. I don't think a lot of people could do that. B, I think and that would not be done by our team, for example. That would be another team that would have to do that. And second, like if you did have a model that could generate these viruses, if those models are all out in the open, you could potentially use those models to screen new sequences and identify outputs of the model as well. So I think on balance, I think there's a lot of benefit in open sourcing the model. Yeah, I think it's in terms of and in terms of where the models are right now, I think generating things at the tens of KB probably within scope, but even generating new life forms or something would require more model development. But I think it is an important policy and regulatory question about if AI can accelerate evolution to design completely new species, how would we deal with these completely novel species? But we can potentially leverage lots of precedent around containment and biosafety But there's maybe some unique aspects to AI generated organisms as well. But I don't want to venture too much into that because we're not there yet, and I don't have like data showing that we can generate AI generated organisms. But I do think that as these technologies improve, we should be having these conversations. But also, I think that as the models get better, like what we can't do in biology is control complex biological systems to cure disease or fight multi-drug resistant bacteria. So I think I'm actually very optimistic about the ability for these models to make a lot of, or to do a lot of good for human biology and human health if they're developed responsibly. So I think that's that.
Question on the clusters you mentioned. I don't understand the technical parts of your genomes, but have you validated how close the clusters are in the latent space? Importance of your inference guidance and experimental validation.
Yeah, in terms of the latent space, we have not actually looked at like the embedding space or anything. We do lots of work in the sequence space or like in the token space. And we can actually see like very interesting sequence changes that would be very hard for like a simpler model to do. And a lot of these sequence changes are actually very novel. Again, we can like compare these sequence changes to natural databases. And some of the mutations the model is making have never been observed in natural sequence databases before. So there is like a lot of novelty in these generated sequences.
Thank you. That was really fascinating. Question on the fine tuning with the phi X sequences. It looked like EVO2, the loss went down much faster than EVO1. Do you contribute that to the eukaryotic genomes? Is there more prokaryotic genomes? And the follow-up question is, if you're going to invest in the generation of new data, where do you see the best value in getting new genomes?
Yeah, I'm not sure why the loss went down much quicker. There's a number of potential reasons. Yeah, potentially there's a lot of eukaryotic data, or EVO2. It was also trained on a lot more tokens. It's trained out for a lot longer. There's probably a lot of like maybe implicit regularization there. So, but I don't want to speculate too much on that question. And then in terms of new data collection, I think so. If you're a biologist and you want to design some interesting or useful new function, if your function is very close to something that's been sampled by natural evolution, then these models will, maybe with not any additional data collection at all, be also useful for that task. But if you're interested in some biological function that's way out of distribution from nature, that's very poorly represented in natural sequence databases, there I can see new data collection being very useful, and maybe you use that new data collection to fine-tune the model or guide it in some way so that it's useful for this completely de novo task, very different from its original pre-training data. So that might be where new data collection would be the most helpful.
I was wondering if you could create new synthetic data by using combining your EVO phages with phi X, like Appelmans or whatever, to kind of force it to create new variants that might work in the lab. You can then like figure that out. What's going on?
Yeah, there definitely. I think you can just have purely synthetic data. There are a few biological experiments you can do that are very high throughput, and the throughput is you can collect like millions of data points with a certain set of specific experiments. So it could actually also be worth like generating 1 million examples with the model, screening all 1 million of them, and further improving its outputs with the fraction that worked and are validated in the lab. So
fascinating work. I'm curious, you framed some of this work with the example of the phage therapy treatment that's being trialed. I'm curious what would need to be true in order for this cocktail of de novo phages that you've created to actually be used in a phage therapy trial. Is that something that people are working on or would be working on? And what would need to be true in order for that to happen? Or are we still not yet there?
I'm actually not a phage therapy person or phage biologist. There are phage therapy people in the audience. You should ask them. But that's like one very exciting possibility. Actually having some of these phages actually help patients could be cool.
Hi, thank you. You mentioned the optimism earlier. I was curious in the process of open sourcing this, if you've already seen any applications or use cases that have particularly inspired you.
Oh yeah, it's pretty. Yeah, I got an email from a team at Yale that apparently used. I didn't talk about this in this talk, but Evo2 is actually great at scoring the pathogenicity of variants to the human genome. Even though it only has one copy of the human genome in it, the evolutionary prior is good enough that it's like useful for human genetic disease. And actually, Evo is state of the art at predicting effects of mutations to the human genome that are associated with Alzheimer's. So that was pretty cool, seeing the Alzheimer's data. There's another lab that we collaborate with at Stanford that has been using Evo to also generate interesting molecular tools and technologies and have been making a lot of progress. So those two stand out immediately, but it is always fun to hear what people are doing with the models.
Thank you for the cool work. Just wondering, like, for all the different ways to explore with like context engineering or prompting for synthetic design, I was wondering if there are smarter ways that don't rely on the natural context of genome, like downstream or upstream for generating, or are there better ways for like encoding if you want a specific function or feed them with like a specific organism you want and then use that latent embedding for generation? And next question is, I'm kind of curious, what's the actual efficiency with the prompting used for like beam search or for other algorithms in your paper? And are there any ways you can speed them up or encode the entire function into the generation process? Thank you.
Yeah, I think you're on to, yeah, it's a really good question. We've done like a lot of low-hanging fruit things to improve, say, the efficiency of the model's generations. But yeah, there's a lot of more complicated things or slightly more complicated things you could do in addition to just using the natural context. Yeah, like explicit conditioning in sequence space, like training like encoders or something to improve the efficiency. The efficiency for beam search is high enough for the chromatin task where we don't need to like explicitly condition the model on chromatin. But for things that are like very rarely sampled by the model, you probably would need to provide some explicit conditioning. So yeah, it's a great question. It's an active area.
Yeah, thanks. So one choice you made in training Evo2 that was interesting was for each species, you have a single reference genome. And one thing I wanted to ask is whether you're looking at architectures to capture within species variation. Like I'm in particular thinking about plant breeding and the extent to which, for example, a different variety might be resistant to a disease. Do you think we need to change the architecture to capture multiple variants of the same species, or is a single reference genome per species enough?
Yeah, I think that it's still very interesting to me how far you can go with representing all the organisms by a single reference genome and it still working well within an organism like humans. But we do think, probably in the plant case, but also in the human case, if you do in the right way provide additional information from the actual species of interest as additional information to the model, somehow additional training, or otherwise, it should hopefully also improve performance on specific tasks.
Just a question about like your intuition for like what comes next. I guess, what do you see the next few years like encompassing in terms of what scales up the most? Is it just like scaling up retraining, or is it incorporating like fast experimental validation loops to do like RL on, or focusing on long context? Just like what do you feel like matters and will be important in the next few years?
Yeah, and in terms of the modeling, I'm very excited about. Yeah, I think in biology we're often interested in achieving very specific functions, and some of these functions are close to nature. Some of them are very far from nature, like very complicated circuits and things like that. And any way of achieving these very complex functions, I think we would be very interested in. This general idea of like, how do we add more conditioning to the model, and how do we control its outputs better, get more reliable predictions. I think pre-training will be a part of it. Very clever ways of post-training the model, or clever ways of using the model just purely at inference time too, are also all of those things will be useful. Yeah.
Yeah, so I'm curious, for a potential end user who really only cares about one species, I'm curious like how your approach would differ from applying the same algorithms, but just to data from one species.
Yeah, and if it's a single, a lot of model pre-training and this whole scaling hypothesis and things like that, like the model improving with scale, I think assumes that the underlying data distribution is like very complicated, and there's a lot to learn from it. So if you're only training a model on a single species, you probably don't want to use models, like you don't want to use like transformers that just and where you scale compute on a data set or something. You probably want more efficient architectures or simpler models. I think, yeah. But again, with the human stuff, the pre-training paradigm can be useful for specific species as well. Like there might be sufficient information learned during pre-training that it can transfer to a specific species very efficiently. But yeah, if you only have data from a single species, then you probably don't want to scale a 40 billion parameter model or something.
Thank you.
Really wonderful to have you kick off this academic year's research seminar. So thanks again for joining.